Dear students, as you know, there are multiple types of RNA secondary structures that can be formed as a result of the folding of the RNA molecule onto itself. In this session, we will see another type of the secondary structure that is formed as a result of this folding. The structures that you know already include helices, bulges, interior loops, and the helices. Today we're going to look at the secondary structure that is called the intersection or the junction. It is a very important RNA secondary structure. So in this structure, the 5' end of the RNA molecule is coupled with the 3' end. However, the RNA molecule is able to do that only after some of these unpaired nucleotide bases are left out. Also, it is called as an intersection because it is formed as a result of several helices that come together and a bulge is remaining outside these helices. Therefore, this is a very important RNA structure because 1, it is composed of the helices, and 2, it has the bulge and therefore is a compound structure. So, to summarize this, junctions include 2 or more double-stranded regions coming together and the formation of a bulge at their intersection, and more so, this structure is a very important secondary structure. Dear students, what happens to these unpaired bases that remain unpaired in the form of bulges? It is a very important area of research and is a very interesting function in the RNA. As you can see in this example, a hairpin loop is given, and of course, as you know, there are 4 unpaired nucleotide bases in the eye of the hairpin. Now just imagine that these 4 unpaired nucleotides, they are able to come together with 4 other complementary nucleotides on the same RNA strand. As you can see, this is exactly what is shown in this figure. This is considered as an anomaly in the RNA folding process and is called as a knot or a pseudo knot. In the next part of this figure, you can see that there are 2 hairpins in red and green as shown here, and that the 4 unpaired nucleotide bases that are here at the center, they come together with the 4 unpaired nucleotide bases in the eye of the second hairpin. So this is called as the kissing hairpins. So this is again another type of the structure that is formed as a result of pairing of unpaired nucleotide bases. Thirdly, if you have a hairpin loop with its 4 unpaired nucleotide bases and it comes together with the nucleotide bases that were uncoupled in a bulge, then they can also make complementary bonds. Dear students, as a result of this is called as a hairpin and bulge contact. Such structures are called as the tertiary RNA structures. As you know, the 1' structure or the primary structure, the 2' structure or the secondary structure, and now the 3' or the tertiary RNA structure has been elaborated. So what happens when the unpaired bases in the 2' region bond? As I just mentioned, RNA tertiary structures are formed. You can just represent them by a 3'. The question that is important to understand here is that the 2' structures were formed in order to impart stability to the RNA molecule. What about the formation of the 3' structures? Does it make the RNA structure more stable? Also, what is the role of these tertiary structures in the biological system?